Now, for the second session, I want to introduce Chris Hine. Please, Chris. Thank you.
Thanks for the introduction. If you all could please get seated, we'll start with the next session. So I'm Chris Hine. I'm an associate professor at Cleveland Clinic, and I'm proud to announce Dr. Michelle Harvey. Dr. Michelle Harvey is a research dietitian at the Prevent Breast Cancer Unit, Manchester University Hospital Foundation, and has been a state registered dietitian since 1991 and a research dietitian since 2001. Her research involves developing and pioneering intermittent low-calorie diets and intermittent fasting. And with that, take it away, Michelle.
Okay, great. It's great to be here. Worth waiting 4 years to come. This fantastic, beautiful place and fantastic facility you have here. Okay. So, my disclosure, I'm a registered dietitian, researcher, and I have authored 3 self-help books to do intermittent diet. All the proceeds go to breast cancer research, not to me. So I'm going to be talking about what I call intermittent energy restriction, and particularly the types of diets where you might do 1 or 2 days of low-calorie intake a week. I'm not talking about time-restricted eating. I'm not talking about fasting mimicking diets. Other people are talking about those. So I'm talking about those diets where you have days in the week that you repeat week in, week out that are lower calorie, not necessarily not eating at all. And I'm going to just go over some of the reasons why they might be helpful, share with you some data on their effects on weight loss and cardiometabolic health, and then really think about how we practically use these diets, if they are beneficial. Do people follow them? Are there any harms, particularly coming from a woman's perspective, which is where I do most of my work? And then to share with you in the second half of my talk some clinical trials we've done in different clinical settings with women. Okay, so why might an intermittent diet be better than just asking people to follow a standard, healthy, calorie-restricted diet each week? Well, and all the statements on here really are aspirational hopes. They're not necessarily all evidence-based statements. So one is these diets might be easier for people to follow. We know that people find it difficult to follow a diet day in, day out. So if you only had to focus on it 1 or 2 days a week, that might be easier. And they may have these beneficial metabolic effects. The fact that you're able to have days of, normally when you diet, if you're asking someone to lose weight, they're restricting their calories by about 25% on a daily basis. But with these diets, you're asking them to, perhaps for 1 or 2 days a week, have a much greater calorie restriction of about 75%, but then eat normally the other days. And is there something about big energy deficits and spells of severe calorie restriction that might have beneficial effects on health? And are they healthy for normal weight people? We know a lot of healthy weight people are actually doing these diets. And the claims are that you perhaps lose more of the right types of fats with these diets than you would standard dieting, that they're really good for maintaining lean body mass. There are so many effects that are hypothesized about these things on the internet. A lot of these things are not, certainly in animal models they stack up. In human studies, a lot of them we haven't got great evidence that intermittent diets are better than any other form of calorie restriction for these things, except perhaps the metabolic flexibility angle. So there are lots of claims. So the first thing is, where I've done most of my work, and actually most of the work with these diets has been done, are they effective for helping people lose weight who have overweight or obesity? And this is an overview of intermittent diets, which are the type I've been talking about. So you've got 1 or 2 days of low-calorie diet a week. This was an overview that was published a couple of years ago, and it's a systematic review. So you can see for short-term studies, and most of these were around they were less than 6 months, around 3 to 4 months, these diets are more effective than a standard energy-restricted diet for helping people lose weight. But over long term, they're no different, and that doesn't surprise me, having worked with these diets for years. Most people get fed up with whatever they're doing over long term. So short term, these diets are slightly more effective than a standard calorie-restricted diet. And most of the studies have been conducted in women. You'll see there that 70% of the people in this review were women. So what about dietary adherence? So how do women find these intermittent diets? This was some data from a small study of alternate day fasting by Krista Varaday's group, and basically you can see here that this is their adherence to their low calorie day. They were supposed to have 3 days of 25% calorie restriction a week. Basically, the premenopausal women were not as adherent as the postmenopausal women or the men to this calorie restriction. And you can see that the premenopausal women's weight loss was not as good. And this is something that we see I work with women of all different ages, mainly with a view to reducing their risk of breast cancer. And this is some data from one of our studies where they didn't have to go on an intermittent diet, but they were given that option. It was a weight loss study where we were just testing different ways of supporting people with these diets. And we had 209 women aged between 18 and 75. And you can see that at the start, 88% of them wanted to follow an intermittent diet. By the time they got to 6 months, only 35% of them were still doing it. By the time they got to a year, only 20% were still doing it. But it was particularly worse in the younger women. And I think that's partly because of the sort of social constructs that younger women have and all the competing things in their lives that perhaps some of the older women who don't have children at home didn't have to deal with. There may be some hormonal, cravings every month, some sort of biological issues. But generally we find young women, whatever the intervention, don't do as well. And you can see that the younger women are more likely to drop out. So I don't think that's a phenomenon of intermittent diets. I think it's a phenomenon of trying to control weight is a problem for younger women. That's something I'm very interested in. But what about harms? Again, you go on the internet, there's all these amazing claims about these diets, but boy, surely you shouldn't follow an intermittent diet. It's going to mess up your hormones. We know that calorie restriction and sort of over-exercise in healthy weight young women can, disturb their menstrual cycles. Usually with around sort of 500 to 800 calorie deficits, you're starting to get effects on luteal hormones and the cycle, and you can eventually become anovulate. But are these intermittent diets where you've got 1 or 2 days a week of a 75% calorie restriction, does that have these effects? And to be honest, we don't have that much data. This is some data from a couple of our studies where we actually just look at menstrual cycle. We haven't done anything clever like looking at their hormones over the month, but we can see that the intermittent dieters, the I-led that is intermittent dieters, and the continuous dieters are the C-led, they have slightly longer menstrual cycle lengths, which perhaps implies a longer follicular phase. We don't know if that's of any clinical significance, but I think there's more work that needs to be done in that area. But overall, I think there's no great evidence that it's disturbing their hormonal reproductive hormones. And the other big concern is, are these diets actually, creating disordered eating? I mean, you're asking people to have days where they have much less calories and then they eat normally. Is that going to create problems? And it depends where you look. So if you look at cross-sectional studies, as this Canadian thing here, if you look across cross-sectional studies, young people, who's doing intermittent diets? The people with some sort of disordered eating or eating disorders. So but the question is there, what direction is it? We don't know whether it's that people who have a tendency to have disordered eating and some psychological issues that lead to that are more attracted to these type of diets. If you actually look in prospective randomized controlled trials where people with overweight and obesity have been asked to follow these diets, actually they have they have improved eating, and they have less disordered eating, and they have reductions in binge eating, and less concerns about body shape, etc. So I think in the setting that they've generally been used for weight loss, they don't appear to evoke issues. And one thing that we've observed, so just to get it clear, these diets are not, if you go on the internet again, these diets are portrayed as periods of feast and famine. So you have your days of calorie restriction, and then the other days you can eat unsolicited amounts of processed, whatever food you like. Well, that's what it says on the internet. The research that's ever underpinned any of these has almost exclusively always advised healthy diets on the unrestricted days. We've always done a Mediterranean diet. In America, it tends to be the American heart. So just to get that into context, but in our hands, when we ask people to do 2 days of calorie restriction and 5 days of a healthy Mediterranean diet, what we find is they naturally calorie restrict on the days when they're not asked to be. They eat about 20% less. So that's why the diet is effective short term, we think. Not only are they getting the weight loss because of the restricted days, but this carryover effect, which is really interesting. So what about its effect on muscle mass? You know, are these diets going to cause a loss of lean body mass, which of course is not what we're aspiring to do? We've done a very short, a small study, very detailed study, quite short term, 2 months of either an intermittent diet, which is 2 days of the low calorie, low carb diet that we have, versus a continuous energy match, continuous energy restricted diet over 2 months. And we followed women up. They had MR imaging of their hepatic and liver fat. We also measured body composition with impedance. And so this is unpublished data. It's something I need to write up. But basically, everything was coming out fairly similar. So they were losing similar amounts of weight, maybe slightly more hepatic fat, but that is certainly not statistically significant. And certainly in terms of losing muscle mass and fat, both diets were fairly equivalent. So it's not detrimental to losing muscle mass. Whenever anyone loses weight, they will lose some muscle mass, but it's certainly doesn't seem to be beneficial either. Okay, so people could do them, but not over long term, and there doesn't appear to be any major health risks. So what about in certain clinical settings? So I think this has already been mentioned. You know, there may be potential benefits for people having chemotherapy, and I know there's going to be more on this later. We were very intrigued. So women that go through treatment for breast cancer tend to gain weight when they go through chemo, which is a bit paradoxical. They tend to gain fat and they tend to lose muscle mass, and they also have pretty horrible toxicities. So we were interested to test our diet, which is 2 days of a low calorie, low carb diet, which ends up being around about 850 calories. It's maintained protein. It's not high protein. It's not low protein either. It's about 80 grams of protein, low carb, 2 days before their chemo, and also in the intervening weeks of their chemo period, which was around 5 months, between 4.5 and 6 months. All they were randomized to a continuous energy restricted diet. We have published this. And so what we showed was the intermittent diet was slightly better for controlling some of these gains in weight and body fat. But what we were really interested in was the effects on toxicity. And I think there's very few studies. In the animal model, again, it's amazingly effective. Most human studies, as far as I've seen, don't really show a signal of improved toxicity. And we haven't really shown it here. Overall, there was no difference this is severe toxicity, a grade 3 or 4 CTCAE. There was no overall benefit. But if you do like a secondary post hoc analysis, which we probably, everybody's always very dubious about for obvious reasons. If you look in the last half of the chemotherapy cycles when they were getting taxanes, there did appear to be less in the intermittent group. Whether that's real, whether that's just a statistical blip. So then, again, not published. This is something I am definitely in the process of writing up. We were very interested to see whether these intermittent diets could not only help prevent some of the weight gain that happens in women, but also perhaps help control the disease. So this was a study in women with metastatic breast cancer. It's not curable. They go, in this example, they were women going through chemotherapy. So they'll start chemotherapy and they'll continue with that chemotherapy treatment until they progress. They have scans every 3 months. We were really interested to see whether the intermittent diet could actually help control the disease process, not just the toxicity, but the disease process, and influence that time to progression. And so you can see it was a phase 2 screening trial. It was designed to have 134 women in it, either randomized to the intermittent diet that I've just described, the 2 days of low carb, low calorie, plus resistance exercise, because I didn't want them to lose lean body mass, and our control was just resistance exercise. So we had recruitment was scuppered by partly gatekeeping in the centres. Nobody wanted to put metastatic patients on a low-calorie diet, and then COVID completely killed it. So we ended up with 68 patients, but when patients were asked, there was a good uptake. And the patients had to have a BMI of at least 25 to enter the study, so the average BMI was around 30. We had a range of METs in the study. They were pretty good at following the diet during their chemo, so the adherence to the low-calorie diet was around, they did 75% of the days that they were asked to do. Again, they were asked to do 2 days a week of this diet. Once they finished chemo, but they were still on the study, it became a lot less. And they were pretty good at doing their resistance exercise, although this is self-reported, so you kind of take that with a bit of a pinch of salt. So the headline is, so the intermittent group did lose a little bit of weight, as you can see here, around 1.8 kilos. But the interesting result, again, it's a small study, it was a one-sided powered test. The median survival in the intermittent dieters until they progressed was 42 weeks versus 28. So it's certainly not doing them any harm. Whether this is, I mean, it could be just an effect of the calorie restriction. We're not saying it's specifically about intermittent calorie restriction, but I think it's of interest. And then just finally, shifting tack, I've also been asked to help with some work with in diabetes. And this is some work we're currently doing in women with gestational diabetes. So this is third trimester, women with overweight or obesity will suddenly be told, oh, you've become diabetic while you're pregnant, which is a bit of a shock for these women. And they're generally told to try and follow a sort of healthier, lower carb diet. And we were interested to see whether this intermittent diet, in this case we've tweaked it a little bit just to make sure it's adequate for these pregnant ladies, is about 1,000 calories, 100 grams of carbs, 2 days a week, 2 separate days, and then 5 days of healthy eating, was better than the best National Health Service care in the UK, which is basically asking them to eat healthily every day of the week. And it was very much a feasibility thing. And in a nutshell, I don't think it's very feasible. We're just finishing the study up now, but these women had to be obese to come in the study. This was their booking BMI at the start of their pregnancy. We had a range of ethnicities, which is typical of gestational or diabetes. So we had a reasonable uptake to the study. A third of women agreed to come in it, but the dropout was terrible. I've never seen anything like it in a diet study. The adherence to the diet, we have just shown you 75% in women going through chemotherapy for breast cancer. It was only 33% here. So it just was not resonating with these women. There are all sorts of reasons. We're doing some quality work to try and unpick that. And finally, something I've been interested in right from the get-go when we started looking at these intermittent diets, could a very moderate 1 day a week of these diets actually try and do primary weight gain prevention, which is really, I think, where we're at. You know, we've got bariatric surgery, we've got GLP agonists, but really, if we can just have something to try and stop people gaining weight, particularly through adult life, could that be helpful? And so I'm in the process of getting my ethics, but we're again looking at the feasibility of a 6-1 diet to try and prevent primary weight gain and see whether it's acceptable, but also whether it's harms. Is it causing eating disorders in the healthy weight young population? Is it causing changes in sleep, menstrual disturbance, etc.? So intermittent diets, they're another tool in the toolbox. They're not an answer for everything. They may have some benefits in certainly in breast cancer therapy. I'm not sure they're a go in gestational diabetes, perhaps for some behavioural reasons, and be interested to see how they pan out. So big thanks to all my fantastic colleagues and the participants who agreed to do these crazy diets at very stressful times. So thank you. Happy to take any questions.
That was great, Michelle. So we actually have time for a quick question, and I'll start it off. So intermittent fasting dietary restriction can often elicit a phase 2 antioxidant detoxification response that's driven by NRF2. So I'm wondering, in your patients with the breast cancer, and you saw a slight amount of toxicity or decrease in toxicity, do they have increased clearance of the chemotherapeutic drugs under the fasting? So does it stay in their body less?
Yeah, really good point. We don't know. So and could that be a detrimental thing Do you think? Or you mean
It could be beneficial.
Yeah, it could be beneficial. Yeah, we actually don't know. And I think I've always thought the question around using calorie restriction around the time of chemotherapy is it could stop, the therapeutic effect of the chemotherapy if you're reducing the proliferation of the cancer cells, which I think people believe it doesn't. But yeah, we don't know. It's a really interesting question. Yeah.
Okay, great. Keith.
You have a lot of experience in enrolling patients in clinical trials. We all fear malnutrition. You seem, if I heard correctly, use a body mass index threshold for enrolling patients, so you're excluding thin patients. In the Lucerne trial, they avoided people that have diabetes that require insulin. We use an albumin in our preoperative patients. What's the right way to determine if we're going to harm someone by preoperatively or, perinatally, etc., restricting dietary intake?
That's a great question, and we don't do it right. So what you really want to do is I think, don't select people who've got sarcopenic obesity. You don't want people with sarcopenia who've got a lot of muscle wasting, I don't think. And so really you need to be measuring some form of body composition, don't you, in patients, I think. And we know that it depends on what you're going to do to them and what they've been through and what they're going to be going through next. But yeah, a cancer patient who I guess has lost a lot of weight, up to that point, you might be suspicious. But I think probably we should be looking more at body we need some easily measured body composition measures. I mean, we've used impedance. They're not that great in people going through various treatments, as I'm sure a lot of us are aware, because of the shifts in intra and extracellular fluid. But I think we should probably be looking more at BMI is not terribly useful. Yeah. But there is a potential for harm, I think, with any of these diets. I think we have to be aware of that.
Thank you.
Thank you.
So I'll call to the stage next Charlotte Mann. Charlotte is a PhD student, PhD candidate, I should say, in the Department of Health Sciences and Technology, ETH Zurich in Switzerland, having been under the supervision of Drs. Catherine de Bach, Sarah Mitchell, Michael MacArthur, and the late James Mitchell. She completed her Bachelor of Science in Biochemistry from the University of Cologne, and then her Master of Science in Biochemistry at the University of Cologne again, in the Harvard T.H. Chan School of Public Health. Her research focuses on sulfur amino acid restriction and its downstream metabolic effects on muscle substrate utilization.
No. Yes, thank you. Sorry. Yes, thank you, Chris, for the kind introduction, and I'm very proud to be representing Jay's Swiss part of his journey before his passing. And I'm going to take a step back from all these patient data, and we really try to kind of understand the basic science that's happening on a baseline when we treat animals with this sulfur amino acid restriction, focusing on endurance exercise capacity. And my old lab, so Jay's old lab, was really interested in dietary interventions and aging, mainly focusing on this sulfur amino acid restriction, which has been around since the early 90s, and it's been described to have a pro-longevity, increase in health and lifespan effects in different model organisms. This paper is focusing on mice, and you can really nicely appreciate that animals that were put on this sulfur amino acid restriction, or they call it methionine restriction, they live significantly longer. And this diet also has a really interesting metabolic effect. That is that these animals that are put on this sulfur amino acid restricted diet maintain a lower body weight throughout the duration of the study, with this interesting controversial actual increase in energy intake. So we really wanted to understand how this loss of body weight and increase in energy intake moves forward on a behavioral phenotype. So just to sum up the dietary intervention that I've been working is, it leaves the overall macronutrient composition unchanged, which is very promising for translational purposes in the clinic. So we have an unchanged ratio of fats to protein to carbohydrates, and we really only restrict the energy intake from the 2 sulfur-containing amino acids. So energy from methionine is restricted to 0.1 to 0.17%, and cysteine is completely eliminated from this diet. And a previous paper from our lab in 2018 showed that this dietary intervention actually also leads to an increase in vascularization. And taking all this information together, we kind of wondered whether or not this overlap with exercise, which is a very prominent intervention to also prevent aging, actually leads to this basically increase in endurance exercise in sedentary mice just by eating a sulfur amino acid restricted diet. To investigate this, we kind of went away from the aging field and just looked in young, healthy, male black 6 mice if 7 days of the sulfur amino acid restricted diet are enough to, first of all, induce the metabolic changes that this dietary restriction includes. So we monitored the body weight and food intake over the duration of the 7 days, and indeed we found that 7 days of sulfur amino acid restricted diet are enough to significantly lower body weight to about 10%, and the mice eat significantly more over a 24-hour period. So after 7 days, we said, okay, our diet works, let the animals run. So the animals underwent a one-time exhaustion treadmill test, which is the forced maximal exercise capacity test. And indeed, we saw that 7 days of sulfur amino acid restricted diet are sufficient to significantly increase the distance that these animals run. So the control group runs around 500 meters. The dietary intervention group, after only 7 days on this diet, sedentary mice, run about twice the distance, so 1K, which is fun to monitor as a scientist. To see whether or not 7 days is also enough to systemically alter the metabolism of the mice, we used metabolic cages where you measure gas exchange, energy expenditure. They also have access to voluntary running wheels, so we measure overall their behavioral and systemic metabolism. And on day 7, we already saw this it's very widely published that sulfur amino acid restriction leads to an increase in energy expenditure after a long-term period on the diet. But already 7 days were enough to significantly increase both in the active as well as in the passive phase in these mice, their energy expenditure. And what we focused on thereafter was actually that after 7 days, and this has not been published, or in the long term this has not been seen actually, that these animals have a decrease in respiratory exchange ratio. And the respiratory exchange ratio is a mathematical measure to infer based on the carbon dioxide produced and the oxygen consumed, which source animals use for their energy production. So the closer this ratio gets to 1, the more the energy production is dependent on carbohydrates. The closer it is to 0.75, the more the animals rely on lipolysis for their energy production. So we hypothesized that this 7 days sulfur amino acid restriction kind of leads to an increase in reliance on lipolysis for energy production, which is also seen in endurance training. So you train your muscles to rely longer on lipids for energy production before you hit your carbohydrate and lactic acidosis threshold. But because in these indirect calorimetric measurements, you can't really see what the animal is using at an exercise time point, with the help actually of Florent, I went to Lausanne, where they have these treadmills that I can run my mice in and use real-time RER measurements, so real-time basically gas in capsule treadmills, and measure the RER during the exercise performance. And when we look at basically the graph that we're going to look at now is these RER trajectories over the time of the exercise. And what you expect in a kind of moderate endurance exercise training is that throughout the duration, about 75% of the duration of the exercise, the animals mainly rely on lipids for energy production. And then you have this anaerobic threshold where you have a steep increase in slope, indicative of the switch to carbohydrates, which is then followed by this break off, which is when the animal is exhausted and burned out. So I did the same exercise test with my animals, and if we just look at the trajectory of ROR over time in seconds, this mainly shows us the sulfur amino acid restriction animals run longer, but it's really hard to really compare what is happening at different time points or different phases of the exercise. So to do this, we normalize each exercise run to its respective duration, thereby getting to a time of from 0 to 1, and this allows us to really nicely see that we have this prolonged lipid utilization phase in the sulfur amino acid restricted diet, which is mainly described by this lowered RER at the max exercise time. So we think that 7 days of sulfur amino acid restriction diet are enough to basically shift the substrate utilization during exercise to a more lipogenic focus. To see whether or not these behavioral changes are also conferred to on a transcriptomic level, we performed bulk RNA sequencing data on 2 different muscle types. So we have different metabolic depots within our muscle types. We have glycolytic muscles such as the EDL or white muscle types that really mainly rely on glucose, and we have oxidative muscle types such as the soleus that really in a normal state really only rely on oxidative phosphorylation. So we performed bulk RNA sequencing after the diet in these 2 muscle types and compared them to this paper that was published late last year, which is really handy, and they basically describe the molecular control of endurance training and on a transcriptomic base in mouse skeletal muscle. And when we compare, focusing, for example, now on mitochondrial matrix genes, our 2 different muscle types, so oxidative and glycolytic, after the diet with the effects that they saw after training, we really nicely saw that the sulfur amino acid restricted diet mimics endurance exercise training on a transcriptomic level, mostly in the glycolytic muscle depot. And we confirmed this on Western blots using just focusing on oxidative phosphorylation complexes. And I'm highlighting here the succinate dehydrogenase B, which is the linker between TCA cycle and oxidative phosphorylation. But in general, you can appreciate that the most complexes are upregulated after the diet in the glycolytic muscle depot and not in the oxidative depot. This tissue specificity due to the diet is also specific to fatty acid catabolism. So if we look at a gene set that is only focusing on genes that are known to be associated with sulfur amino acid restriction, so we have cystocyanine, CTH here on top, and other known genes that are associated with to be respondent to sulfur amino acid restrictions such as ATF5, etc., we don't see this muscle specificity. So we really think that the sulfur amino acid restriction diet is sufficient to lead to an oxidative shift in the glycolytic muscle depot. To see what's happening on a systemic level on lipoic intermediates, I had a chance to visit Mike and Sarah in Princeton, and we performed these circulatory flux measurements. So turnover rates pretty much describe whether or not you have an increase in production or consumption of a certain metabolite. And you can do this by using catheterized mice, and we put them on the sulfur amino acid or control semi-purified diet for 7 days, and then infuse stably labeled linolate until it reaches steady state, and then measure the circulatory flux. And 7 days was enough to significantly elevate whole body turnover rate of stably labeled linolate. And when we wanted to, this basically just tells you that the whole metabolism, the whole animal kind of increases this turnover rate of this metabolite. So we wanted to see whether or not this is due to increase of uptake or utilization in our muscles. So we performed beta oxidation ex vivo with labeled palmitate, and we could see that after 7 days of the diet, the EDL, so the glycolytic muscle depot, significantly increased their beta oxidation of palmitate into deuterium water. But since basically the flux, if you want to look at the flux and want to tell a story based on this, you also have to look at the overall concentration. So the lab in Princeton really likes to use this metaphor of a freeway, where you can say, you need to look at how many cars are on the road to really know whether or not you have just less cars that drive faster, more cars that are slower, or the same amount of cars going on and on and on. And so to test to see whether or not our increase in overall turnover rate was just due to an increase of cars, so an increase of concentration or pool size. We performed different measurements, metabolomics, looking at the free fatty acids, as well as lipidomics, but also intramyocellular lipid droplet staining. So I'm going to show you this. Overall, the results were all the same, and that is that we don't have a different storage or concentration of lipid species within our muscle depots. So we think that it's really, so we were wondering if we don't have an increase in concentration or mass action based on increase in concentration and thus flux, what else could be driving this increase in turnover rate? And one road we took is, do we have an increase in transport of lipid species into the muscle? So to investigate this, we created a mouse model that is crossing the CD36 flux animals with the PDGF beta Cre, which is a Cre driver specific for endothelial cells. And CD36 is a fatty acid transporter such as GLUT4, which gets translocated after stimuli to increase fatty acid transport into the, in this term, into the endothelium and thereby into the tissue, the respective tissue. We confirmed our knockout efficiency looking for in different tissues, so in muscle or in brown adipose tissue, we saw a nice knockdown of endothelial cell CD36 double positive populations. And when we looked at the effect of the diet, we don't really see an effect of our knockout looking at body weight loss. So we see a same amount of about 10% body weight loss independent of CD36 being present or not. Then I went on and performed our one-time exhaustion at treadmill exercise test, and we saw a slight attenuation of our running phenotype. Overall, I have to say these animals, all of them ran very poorly. I think the tamoxifen induction of this Cre really affects the running behavior of these animals. But we do think that we see a slight attenuation after the knockout of CD36, but it doesn't explain the overall running phenotype. So circling back to the beginning of the talk, and I briefly touched upon this paper that we published in 2018 showing an increase in angiogenesis. So if you go back to the freeway metaphor, basically, if we don't have more entering onto the freeway, do we just have more freeways? So basically, do we just have increase in vasculature in the muscle that's driving increase of accessibility of lipids that go into the muscle, driving our running phenotype? To test this, again, I put young mice on the sulfur amino acid restricted diet, and I co-treated them with a pan-VEGF receptor inhibitor, axitinib, to see whether or not inhibiting VEGF signaling, which is the main driver of angiogenesis in the muscle, leads to a prevention of running phenotype. We don't see an effect of this treatment on our body weight of the diet, so the diet itself works fine, even if we inhibit VEGF signaling. But when we look at running, actually we see complete attenuation and diet and treatment interaction effects after inhibiting of after inhibiting of VEGF signaling. Though, when we then wanted to confirm this by looking at increase in angiogenesis, so increase in endothelial cells, we actually don't see an increase in endothelial cells after 7 days, which is, we would have been very surprised if 7 days, so in a physiological sense, 7 days would have been a very short stimulus to already increase neovascularization in the muscle. So we kind of looked in the literature and wondered what other non-canonical pathways are there that are associated with VEGF signaling. And that is actually quite interesting because some papers published in 2010 say that VEGF-R signaling leads to an increase in fatty acid uptake into the endothelial cells. So to test this hypothesis and kind of already coming to the end of my data, what we did is we again used catheterized mice, put them on the diet, and then we infused them with the drug for 24 hours to reach steady state, and at a concentration where it's not harmful, but where the IC50 of the two VEGF-R receptors are being hit. And then again, we measured the turnover rate of the fatty acid linolate. And we think that inhibiting VEGF receptor signaling is mainly preventing our running phenotype by inhibiting this increase of circulatory flux of linolate. But I'm still working on basically finding the hard evidence to support this hypothesis. With this, I do want to come to an end, and this is a very busy slide. I'm just going to briefly summarize it. So after 7 days of sulfur amino acid restricted diet, we see an increase in these oxidative phosphorylation genes, as well as CD36 and LPL, which I haven't shown yet. Didn't show you the data today, but also an increase in VEGF-A, B, and the respective receptors. And I'm currently in the last few months of my PhD trying to, not being on Zoom, trying to understand how these pathways interact, and yeah. I can just go to the last slide. Where's the mouse? So I want to see how these different pathways interact to basically, in the end, lead to this push of subset utilization switch during exercise, which allows prolonged running in male sedentary mice after 7 days of sulfur amino acid restricted diet. And with this, sorry for this brief interruption, I really want to have a few people to thank, mainly Jay, for taking me in Boston and then moving me to Zurich. After his passing, really Mike and Sarah, who can't be here today, they have more important things to do now in their life. But without their tremendous effort, this project wouldn't have been able to be close to finish after 4 years. And yeah, I'm happy to take any questions.
That was a great talk, Charlotte. And I know Jay would be very proud and happy with all the progress and all your accomplishments. So I have a quick question. We have time for maybe 1 or 2 questions. What about lactate? Have you measured lactate levels in these mice when they're at the kind of maximal exercise?
So we actually did that, and we had different outcomes. So when we first did it, the lactate was lowered at exercise, but so we didn't have a really nice setup. So we did tail vein, just like the glucose meter for lactate. So it's super variable, it's super dependent on the person who is handling the animals. I did get the chance when I was in Princeton to also measure lactate flux, and it seems to be decreased. So the lactate utilization apparently goes down. But yeah, we would have to repeat all of these experiments to be confident to state that.
Great. Yes.
Very nice talk. Did you look in the EDL? The EDL is the mixed composition is not purely glycolytic, standing very good. So did you look the muscle fiber types?
So yeah, actually that's a good point. After the bulk RNA-seq, fiber type switching was one of the main hits that we got. And for example, myosin heavy chain 7 goes up in the EDL after the diet. But when we look whether or not this is conferred to on a fiber type switching level, I don't see a switch yet. But I think this could also really be due to the timing, because 7 days is really short to have really like fluorescently based imaging picking up these changes, I would say. So I think it could be, I'm not excluding that it's happening, but I think if we would wait a longer time, then we probably could see it.
Thank you very much, Charlotte. Up next, we have Dr. Nezilin Alkis. She's on Zoom. Okay. So while we're getting her Zoom set up, I'll introduce her. Dr. Alkis is a professor doctor specializing in anesthesiology and intensive care, and chairs the Department of Anesthesiology and Reanimation at Ankara University Medical Faculty. She graduated with her medical degree and trained in residency program at the Ankara University Medical School. She has served as the General Secretary and President of the Turkish Anesthesia and Reanimation Society. Her research focuses on the ARES protocol with preoperative carbohydrate loading. And when you're ready, take it away, Dr. Alkis.
Hocam sesimiz geliyor mu Neslihan hocam? Bizim ses açık mı? Hocam biz başlık sisteminizi sesinizi açıp. Evet hocam, evet hocam. Lütfen. Lütfen. Tamam. Evet hocam görünüyorsunuz. Hocam geliyor mu sesimiz? Hocam sizin sesiniz bir saniye hocam. Sisteme bağlı mıyız?
Recording in progress.
Evet Atacığım.
Hocam bir ses sorunu var galiba ama.
Nasıl?
Şimdi oldu galiba. Kapıyorum telefonu.
İyi mi?
Geliyor mu hocam sesimiz? Geliyor mu hocam sesimiz?
Geliyor. Sizin
Hocam
sesiniz-
merhabalar.
Geliyor benim sesim.
Şu an geliyor hocam. Kamera görüntüsü aktarıldı mı? Kaç taraf?
İyi mi şimdi?
Hocam iyiyiz şu anda ses geliyor. Toplantı salonundayız. Sayın Rektör hocam buradalar.
O zaman başlıyorum.
İzniniz olursa.
Çok teşekkürler. Başlıyorum Atacığım.
Biraz tamam. Volümü biraz arttıralım mı? Hocam
Hello
lütfen.
everybody and dear chair, dear colleagues. I want to begin with thanks to Professor Zafer Türk and all the organizing committee. And this is a very big honor for me and I'm very happy to be a speaker in this such a special meeting. And I'm speaking from Ankara, unfortunately, but I'm also very happy to be here with you. Okay. My lecture is about perioperative optimization for the surgical patients. Where I am from Ankara University and Medical School. I'm in the middle of the Ankara with two campuses I'm working in. These campuses have almost 2,000 beds. And annually we perform 30,000 annually surgeries, including major surgeries. These are transplantation surgeries, cardiovascular, and also oncologic surgeries. And what I'm talking about this morning, I will mention about some surgical patient population and what is surgical stress, and its trademark ERAS, Enhanced Recovery After Surgery Protocol. I will mention about it, and the point is what is the ERAS protocols related with carbohydrate metabolism and carbohydrate loading. When you come to surgical patient population, we should understand that this population is also public health issue for our healthcare strategy. Because there is a tremendous volume of worldwide in all settings, both rich and poor countries. And general population is changing very fastly nowadays. Life expectancy is increasing. Elderly patients are growing and growing. Obesity and diabetes are the main concerns currently in the general population. And what about surgical patients? They are very different from the last decade. And in the near future, we know that a big portion of these patients are obese patients. Age also, frailty are all surgical risk factors for these patients. And what is the consequences? Wound healing capacity, cardiovascular compromises, immune system compromises, and we need urgently high quality and safe surgical care measures for this public health level. Okay, let's talk about some numbers. When we come to surgery numbers globally, almost 300 million major surgeries are performed. And the point is, mortality is very important for the surgical patients. As you see in the table, almost up to 4% will be dying. And the third line is the major surgery for the death reasons in the world. These are for, as we looked from our perioperative care, we can reduce mortality, we can prevent morbidity. And it is beginning from the surgical stress. What is surgical stress? Surgical stress is the acute response to body. It begins from the incision of the surgery, from the sterile injury, and simultaneously induction of anesthesia with synchronizing anesthesia performing, and it's increasing the sympathetic response. What is the endpoint? Inflammation increases, coagulation compromises, immune system compromises, and also susceptibility to infection. And the endpoint is reduces the oxygenation to all the tissues. That's the main point. And the secondary injury, by reducing the tissue oxygenation, all the organs can fail. Brain, heart, lung, kidney, they are all in danger when it is the subject of surgical stress. The stress is not a new topic for perioperative care. In 1913, Dr. Charied defined the shockless operation. It means that can we perform some surgeries without shock, without surgical stress? Yes, in that time, in the beginning of 20th century, Dr. Cannon said that it's with the aid of adrenal secretion. It is with the aid of acute response, which is hypothalamus, pituitary, and adrenal cortex responses. And it was the beginning of the stress surges, but also in the 20th century, in the history, we are all seeking for the high surgical risks. It's on the muscle, it's on the protein and postoperative functions. It's on the mainly metabolic preparation before surgery to improve outcomes. A striking point is we are still trying to these metabolic preparations before surgery to improve outcomes. And we know that metabolic preparation is feasible and effective nowadays. And what are these perioperative complications? As we said before, hemodynamic compromises and hyperinflammation, metabolic imbalances, and hyperglycemia. And the point is, it's a surgical stress response is a widespread changes in organ function. It's mediated by trauma-induced endocrine metabolic and some hormonal changes. And it's also cytokines in this cycle, active components, interleukin-1, interleukin-6, and TNF-alpha, stimulates the HPA axis, and the catabolic phase begins. It is beginning with the cortisol, catecholamines releasing. And the endpoint is insulin resistance. And the key point for the stress response is the insulin resistance. We all know about insulin resistance, what is it? But the consequences, the endpoint is loss from muscle, loss of muscle mass. And it is the point that reducing muscle function, because they can take glucose into the cells of muscles. And these changes are, yes, temporary, but it's for a long time, at least 4 weeks, even after an uncomplicated colorectal surgery. And as a result, we can say that insulin resistance and hyperglycemia are very closely associated with the perioperative, it means postoperative complications. Yes, as a summary, we can look to what are the endpoints, what are the clinical points. Pain, if the patient is with pain, it's a complication. Gastrointestinal paralysis, cerebral function, dysfunction, nausea, fatigue, and all of them are the components of prolonged recovery. And let's come to ERAS. It is the acronym of Enhanced Recovery After Surgery. We are trying to make the recovery, surgical recovery, enhanced, quicker, and qualified. And what is postoperative recovery? It means that we need gut function return as fast as possible. We need mobilization as soon as possible. We need painless postoperative recovery time. These are all very much related with insulin resistance, and these 3 aims should be covered for enhanced recovery. Yes, when it comes to ERAS, and in this figure, I can summarize that it's evidence-based, it's multimodal. It's about surgery, it's about metabolism, it's about dietary functions, it's about physiotherapy, it's about anesthesia. And it's evidence-based, multimodal, and multidisciplinary initiative to improve the perioperative care. In perioperative period, we have such an item that perioperative carbohydrate loading and perioperative fasting strategies. What they do as a result, they make the metabolic stress reduction. And the key factor in this postoperative recovery is to minimize the stress of surgery. Okay. Of course, it is the same terminology with insulin sensitivity. In that figure, we can see that in the traditional care, the metabolic stress and insulin resistance can reach with a long for days and weeks recovery because of preoperative overnight fasting, because of no nutrition, because of low caloric fluids during surgery, preoperatively, postoperatively. But in ERAS care, if we can manage it with a good dinner, a very good sleep before the night of surgery, and carbohydrate treatment before the surgery, and thoracic epidural management for the pain management. And after the surgery, immediate feeding, immediate mobilization, it will take a very short time for the insulin sensitivity to have for the postoperative patient. And ERAS protocol is a chain of events during the patient's surgery. Surgery begins from home to the home, but it's time in the clinic, it's in the pro period, it's in the intraoperative, it's in the ward, it's in the home. The recovery is a total teamwork. Who are they? Surgeons, anesthetists, intensive care specialist, dietitian, physical therapist, and medical doctors also when necessary. Earth Society protocols are for some special surgeries. In the beginning, it was with colonic resection. As you see, the recommendation, the element is from midnight to preoperative 2 hours before anesthesia. We don't want meal per hour. We're just reducing fasting time. And we give carbohydrates, clear fluids to our patients. And in the postoperative period, we should control glucose immediately as possible. And there's, Earth Society has not only for colonic surgery, as you see in the time, and it's currently a lot of different surgeries, major surgeries. There are guidelines, recommendations, evidence-based recommendations from ERAS Society currently, and we are following as ERAS performance these guidelines during our surgeries. And let us come to carbohydrate metabolism. What is the relation between the surgery and carbohydrate metabolism? As all we know, insulin is the main anabolic hormone and essential for all the metabolism. But in the fasting time, metabolism can go through an catabolic phase. But in the fatting time, it will go through with the anabolic phase. We don't want gluconeogenesis, we don't want lipolysis, we don't want proteinolysis, we don't want hyperglycemia. And if you put your patient in the fasting, a long time fasting status, they will all come through what we don't want. When we come to, again, metabolism, also some pro-inflammatory interleukins, IL-6 and 1 cause also insulin resistance. And, but when we feed the patient with up to 2 hours period to surgery, we predominate to restoring glycogen, increasing muscle uptake of glucose, and elevating insulin. And the main point is we're halting protein catabolism. How do we do it? In preoperative fasting, as all know as surgeons, it's a big rule in worldwide, but it is not the true rule, because we should try to make, as anesthesiologists, to make an empty stomach for the induction of anesthesia. But we all know nowadays that 2 hours before anesthesia feeding with clear fluids do not increase the gastric volume. And on the contrary, intake of clear fluid with the carbohydrates, it stimulates to empty the gastric volume and also reduce the gastric volumes, and also less insulin resistance. And what we do is we give 4,800 milliliters of carbohydrates, depend on maltodextrin, 12% maltodextrin drinks on the day of the surgery, before the night of the surgery, and 2 hours before, it's given in the 2 or 400 milliliters of carbohydrates we are giving. What we do is, the purpose is, as we declared before, and some anesthesiology and intensive care societies declared the fasting times and carbohydrate loading, or oral carbohydrate loading. Nowadays, only the European Society, SAIC, is recommending strongly oral carbohydrates. But Cochrane reviews that there is no risk for any case of aspiration. Yes, for every surgery, in ERAS recommendations, that's colorectal, gastrointestinal, pancreatic surgery, liver surgery, bariatric surgery, there are recommendations. And what are the fluids? It's usually the same amount of maltodextrin in the products. And as a very quick review, I will show you some meta-analysis and literature review that support our oral carbohydrate loading. And we know that in this review, but by all the linguists from the ERAS Society's presidency, overnight fasting is very, very special for surgical response, stress response, and we need to give the patient oral carbohydrate before surgeries. And in the other review, we can see that postoperative mortality and can be in postoperative length of stay and bowel functions and patient-related outcomes can be easily handled by these administrations, oral carbohydrate administrations. And in another review, hospital stays can be decreased by this administration. And what about different surgeries? One-day surgery, as a conclusion, it seems that it is not so much supported to give for one-day surgery. We should need to have more randomized control trials for this. But for cardiac surgery, we know that inotropic drugs, ICU length of stay, articulamptic durations, and insulin therapy postoperatively can decrease with this administration, with this protocol. And when we look to a new meta-analysis, and it's a Bayesian network meta-analysis, yes, it's important. There is effective result for oral preoperative carbohydrates, but we need some more studies for the supporting evidences. And in bariatric surgery, ERAS recommends strongly for carbohydrate loading. Yes, there is no aspiration risk for this surgery. As a conclusion, traditional surgical care is really associated with marked metabolic reactions. The point is insulin resistance and the protein losses. But modern multimodal care principles, as from employment of ERAS, we can avoid the side effects. Administration of oral carbohydrate drinks when given up to 2 hours before surgery have been demonstrated safe and effective. And it influences on a wide range of perioperative clinical outcomes. As a result, metabolism and nutrition are backed in the surgical arena today. And thank you for your attention. Thank you for your listening.
So we only have time for one question, if anyone has a question.
One question, yes, please.
Is carbo loading still valid if the patient is diabetic?
Actually, I can't hear easily. Can you
Sure.
help me about the question?
Yeah, I'll repeat it. The question was, if I get it right, is carbo loading prior to surgery still effective if the patient is diabetic?
Sorry. Anybody can...
Please again.
If I was asking if the carbo loading still valid. Carbo loading. Loading.
Carbon loading?
Yep.
Yeah.
Is still valid if the patient is diabetic?
The question is about the carbon loading.
yes, carbohydrate loading, okay.
Oh, yeah.
Is it still valid? Is it still accurate if the patient is
Is it
diabetic?
still accurate for the patients?
Patients with diabetes.
Patients with diabetes, yeah.
No, we are talking about without diabetic patients because we need more randomized clinical trials for diabetic patients and insulin resistance patients yet. Because some literature says that it's safe to give them. Some of them are not very sure about for the diabetic patients. Yes.
Thank you.
Yeah.
A round of applause.
Thank you for your presentation. And
So up next, we'll have, I welcome to the stage Dr. Emel Ergul. Dr. Ergul is Professor of Medical Biology and Genetics at Kocaeli University, Faculty of Medicine. Dr. Ergul graduated from Kocaeli University Department of Academic Data Systems and earned a PhD studying manganese superoxide dismutase gene polymorphisms for breast cancer. So when you're ready, take it away.
Yes, I can. I will open my video first. Evet, öncelikle Zafer Hocama bu fırsatı bana verdiği için teşekkür etmek istiyorum. Ve hepinize hoş geldiniz diyorum. Şimdi Zafer Hoca ilk beni aradığında bu konuşmayla ilgili hocam dedim hani bir fikirlerim var, bir sürü datam var ama hepsi teoretik ve hiçbirisinde hani bilimsel bir şeyim yok. Hoca da sağ olsun şey dedi, sosyal bir sunum yap o zaman bize. Umarım size ilham olur sunumum. Yani direkt bir bununla ilgili direkt bir çalışmam yok. Ama böyle bir genel bakış açısı vermek istiyorum. So, welcome. I first would like to welcome all of you to our country, to our city, to our faculty. Today, what I'm talking about is actually not the topic, it's my dream. And I would like to inspire you in some topics related to lifestyle factors. So my way to this topic started when I was at first class, as a first class biology student. I was fascinated from the cancer pathways. And then I went to the academy to search this topic, and I work a lot on this topic, and I follow the very basic medicine searching rules to understand the diseases. But because I'm not a physician, I don't have access to the patients, I can only make the basic part. And my career was all on how to understand the cell at molecular level, to understand the gene, the genome, and all these things. But later in my career, I asked this question for myself: what was the external factors who provoked the cell to follow the cancerous mechanisms? And first what I started was nutrition and food, and then nutrigenetics, I did a lot of search in that area. I have 2 publications regarding these topics, but it was not enough for me. So I say I should, it's something else that can explain how the human, how the cell is following this pathway. And at the very same time, I learned the battle strategy and a course of leadership, as a course of strategic leadership. And this battle strategy led me to create a sustainable health formula and classify the lifestyle factors into 5 main issues that I want to present to you today. The videos I use are all from social media. I couldn't find some scientific page for it, so I took the pictures from the visuals from social media. And what was my formula? I started with posture, so correct posture. As you see on the picture, the correct posture I couldn't see when I'm out and think people. And from here, I wonder if these other deviations are related to diseases. Because I could not see really the right posture. So, when you don't have right posture, your lung is not functioning properly. And what happens in the
Red
cell?
blood cells travel throughout the body, allowing hemoglobin to deliver oxygen to the cells that need it. Within the cell, oxygen is utilized in cellular respiration, which is carried out by proteins of the electron transport chain in mitochondria. The flow of electrons along the electron transport chain releases energy. This helps drive hydrogen ions across the inner membrane of the mitochondria. Hydrogen ions then flow down their concentration gradient through ATP synthase, driving ATP production.
So, this is what we call normoxia, and what is happening when the cell have normal oxygen levels. But if we could not get enough oxygen, so this started to going to
red blood cells travel throughout the body, allowing hemoglobin to deliver oxygen to the cells that need it.
The very
Within the
first
cell,
step of
oxygen is utilized
inflammation.
in cellular
So I'm not
respiration,
going to talk about this because all of you know very well the other part. And the second part was exercise. I make a lot of public conferences, and I was teaching about this topic, and I was explaining if you do not exercise, your liquids, meaning blood and lymph system cannot work properly. And your cell doesn't have enough oxygen, enough food again. So, about this topic, there are really very much papers, so that means it's time to move. we should, it's not optional according to me. We have to arrange somehow to move. And the other part of physical activity is neuroplasticity. So if you are not moving, we cannot learn properly also. Neuroplasticity means when you learn, your neurons make new connections between them. And the other part for exercises is the molecule secreted in muscles. As you see, there are very huge amount of data about this. So if you lose your muscles, you lose everything. And the third step of my formula is good eating habits. I don't want to say healthy habits because we still don't know what food is healthy for everyone. So one food is healthy for me, but it's not healthy for you. So healthy food, to say healthy eating, healthy food is not very good for me. And I make good eating habits, and just I put very physiological part of the food, meaning mastication, which is the very, very important step of digestion. If you don't have good mastication, you cannot digest the food later in the digestive tract. Eating according to circadian rhythm, and that means when you eat. That is very, very important. At what time you have your food. Prefer natural food. So it's according to what is this. Also, you should choose your food according season. And food grows in local destinations. So the very big problem is now that we can have food from everywhere of the world, and we don't know who is local, who is seasonal. So we have to make some work about this topic also. Fasting is the other issue of the lifestyle factors, and fasting is also it should be fasting programs should be made from the medical professionals, according to me. And the other part is nutrogenetics. What we eat is really according to our genes. So everybody has his own diet, and they should have courage to find what food is good for them. And about this topic, I have a lot of protocols created for me, and I work with other groups on them, and if somebody is interesting, I can share with you. The fourth is breathing exercises. So as you see, it's again related to your spine and to your posture, and again to oxygen, of course. The right posture, and you have in parallel, you see the diaphragm and here, but when you do not have, when you have improper breathing mechanism, so again deviation. And I didn't find some words about this. But what happen again when we don't have enough oxygen. Fatigue and even stress on your heart, elevating cortisol, weight gain, mood swings, and trouble sleeping. Shutdown of the executive functions of the brain, and you can forget about great decision making or real focus. And the 5th step is sleeping. And what I heard in public, and all my students are not sleeping at the time they should sleep. They sleep after 2 or 3 o'clock. That means your melatonin levels, your melatonin synthesis is stopped at that time, and you are going to bed. So what happens when you go to bed at that time? Your food for the brain is sleep, and when you don't sleep properly, you cannot control your thoughts, and your neurons cannot work properly, of course. So that was my 5 steps. I make here very only hints about it. There are a lot of literature, a lot of scientific publication in that area, but they are all at a basic medicine level or biological level. So still we do not have this in the medical education programs, and we should put them to the medical students program. And the other thing that is dependent on sleeping is our circadian rhythm. So the good sleep is between 10 and 3 o'clock actually. You have to sleep very deeply at that time to be sure your melatonin synthesis is at proper levels. And all this you see here, according to these hours, for example, here is hour for physical exercises. It's good at 5 o'clock. So it's good to go to gym at 5 o'clock to get the all benefits from sport. But what sport you should do is again genetic. You should find from which sport you can benefit. Your body, your muscles, your genetics, all these are related to you. Okay. And the other issue of sleeping is your hormones. So it's not actually emotion, it's hormones that are make you feel anxiety or feel depression or feel low. So to have good levels, to feel low, we have to arrange our hormones. And I search for Nobel Prizes. I was wondering if there is something to my topics. And I was very surprised to see that actually all topics were related to my formula. The Nobel Prize in Chemistry 2015, the team take the prize for mechanistic studies of DNA repair. And I read the papers of Aziz Sancar. He is opening with this, he is opening the era of chronochemotherapy. So that means you can do chemotherapy according to your DNA repair. DNA repair is too much during the day, so the day is not good for the chemotherapy. But this is only, again, on only basic science level, so it should be arranged later according to this knowledge. And Nobel Prize 2016 for Yoshinori Ohmitsumi for his discoveries of mechanism for autophagy, and this is what he's talking about it today. It's fasting mechanisms in the how cells work during the fasting. The other is in 2017, the team had the Nobel Prize for their discoveries of molecular mechanisms controlling the circadian rhythm. So for circadian, I mentioned circadian rhythm. And in 2018, the Nobel Prize team had it for their discovery of cancer therapy by inhibition of negative immune regulation. And this is also the way to open for new immune therapies in cancer. And in 2019, the team was awarded by Nobel Prize for their discoveries of how cells sense and adapt to oxygen availability. So as you see, all are on basic medical level, basic science. We have enough data at least to start from somewhere. And the other issue I would like, because I'm biologist, I would like to say that we are very part of the nature. We are human beings, but at the same time, we are Homo sapiens. So we are designed according to nature, and we have to obey the rules of the nature. And here is Homo sapiens, so we have some traits that are with all these animals, but the variation is between species. So as human being or Homo sapiens, our genetics is very unique. So everybody has their DNA is, everybody's DNA is very unique, and all the topics I mentioned are very personal. So and they all topics should be personalized.
Dr. Ergo, one more minute.
What?
One more minute. No, one more minute left.
Okay, yeah, I'm finishing. So there is a lot of data, and it is the era of translational science, so we have to make a collaboration with very different disciplines, not only biomedical sciences. We should make we should make teams with also the other, with sociology, with psychology, and other disciplines that are not in medicine. So this was my data mining actually, and I use very much sources. All scientific textbooks, all scientific database related to biological and medical research, and social science databases, traditional medicine books from different cultures, articles, videos, social media, books and articles about nutrition and fasting, articles, videos, social media, all kinds of literature, novels, philosophical books, and historical and so on. And as you see, I even use battle strategy to create this formula. So it's very interdisciplinary to work on human being. So, and I would like to thank, first of all, to my nuclear family, my extended family, all my close friends, and all the people with whom our paths crossed in my life for all their support and help. All my teachers and mentors and colleagues who paved and enlightened my path in my professional life. The dedicated scientists who I do not know personally, who contributed to this speech with their scientific studies. The technical team of Kocaeli University who helped me place the visuals at my slideshow for their gracious contribution. Thank you very much for listening to me.
Thank you very much, Dr. Ergul. So we are running a little bit behind now with lunch. So if you have any questions for Dr. Ergul, I think we can ask her during lunch. You can right?
Yeah,
they can
yes.
ask you during lunch. Perfect.
Better for me.
So I'll ask Furkan and stuff in terms of us being behind. What time would you like us back to kind of get a little bit more on schedule? Like 13?
I think we should aim for 1:10.
1:10?
Yeah, it's got a 30-minute break.
Okay.
1:30.
1:30. Okay. Okay, 1:30 be back, please. Thank you.